Microorganisms, Classification and Diversity Unit, Video 1. Importance of microorganisms. Bacteria that cause the Black Death, also known as the bubonic plague, and diphtheria, all cause so many deaths that some say microbes have altered the course of history. And we can see the spread of the bubonic plague from 1347 to 1351 and how it spread outwards. Microbiology is the study of organisms or microbes. Microbes decompose dead animals and thus recycle essential elements such as carbon, oxygen, sulfur, nitrogen. Uses of microbes. They are used to develop antibodies, used in the dairy industry for cheese, yogurt, buttermilk, and they are used to study metabolism, photosynthesis, enzyme action, and gene action. And their use in the medical research is very promising. The kingdom Archaea. Archaea bacteria. So it's down here. These are unicellular and prokaryotic cells. So they live in environments without oxygen, in environments with high salt concentrations, high temperature, or high acidity. These conditions existed when first life formed on Earth. So these are really our prehistoric bacteria and kingdom. Our kingdom of eubacteria or bacteria, these are things found in unicellular and prokaryotic. So similar, but are the largest group of monorans. They are microbes that traditionally are recognized as bacteria. They include bacteria and simple photosynthetic, meaning they get their energy from the sun, photo, and chemosynthetic microbes, and the chemo, meaning energy from breaking down chemicals. Characteristics of bacteria. Bacterial cells. Bacterial cells are prokaryotic, single-celled. Their cytoplasm contains no membrane-bound organelles. Bacterial cells have a single chromosome. Most bacterial cells reproduce by binary fission. The individuals of both kingdoms are similar in shape, size. However, it's important to note that Archaea bacteria live in harsh conditions where bacteria would not survive, including habitats with high salt concentrations, high temperatures, or high acidity. So these environments are thought to resemble prehistoric times. Bacteria are classified according to three basic shapes. So they could be spherical in shape, which is known as cocci or coccus. Coccus is meaning just one of them. So we can see the spherical shapes over here. An example of this would be Streptococcus, mutants with tooth decay, form in chains, or Staphylococcus aureus, bacteria on the skin that forms clumps. Rod-shaped bacteria are known as Bacillia or Bacillus, and spiral-shaped are Spiralia or Spiralium. After divisions, they may form pairs, clusters, colonies, or chains, but they are still single-celled organisms. Modes of respiration. So you have obligated aerobes, and these require oxygen for respiration. For example, that would be tuberculous bacteria. Then we can have obligated anaerobes, and they conduct respiration without oxygen. And then we get fecundative anaerobes, which prefer environments with oxygen but can live without oxygen. Most bacteria fall into this category. Modes of nutrition. Autotrophs are few bacteria produce their own food through photosynthesis. We also have heterotrophs. Most bacteria obtain their nutrition by consuming other organisms. We have two types. We have parasites. They live on living organisms, example, disease-causing bacteria. Or we have saprophytes that live on dead and decomposing matter. Reproduction. Asexual reproduction, called binary fission, or the splitting in two. It goes with the DNA is going to double, so your genetic material inside your cell will double. Then the bacterium produces a cross wall in the center, and then two identical cells are formed. So let's look at this a little. So binary fission. So we start first with our single cell organism. It is a DNA ring, a cell membrane, a cytoplasm. So the first step is the DNA has to double, so that's why we see two rings. Then we have a cytoplasmic bridge that forms. You can see that here and here. And your two rings will go on either side. Then the bridge will completely collapse and form a wall, and then we'll have two identical cells. Type is sexual reproduction, called conjugulation. It occurs, however, it is not common. So here we have two mating types, referred to as a donor-recipient. Make cell-to-cell contact via a cytoplasmic bridge. The plasmids, or the small ring of genetic material, are transferred, and the two cells separate. So if we have a closer look, we start out with a donor and recipient. So we have your donor recipient. Then we have a cytoplasmic bridge that forms, but it doesn't completely form. It doesn't completely close. So it allows some of the DNA from the donor DNA to go into the recipient DNA. And then it joins together. And the donor is left with no DNA, because it's left all by itself. Then the cytoplasmic bridge closes, and we have a new cell with new DNA, with your donor and recipient. And then we have our donor cell that dies. It's kind of a counterproductive reproduction. The next one is endosperms, and they form inside a bacterial cell during unfavorable conditions. Endospores are dormant or resting cells that contain genetic material encased in a thick, resistant cell wall. They are resistant to heat and cannot easily be destroyed. When favorable conditions return, the cell wall breaks down and the bacterium begins to grow. Beneficial bacteria. So we have decomposers of dead plant and animal material. They produce a production of acetone, vinegar, lactic acid, cheese, buttermilk, yogurt. They're also an important part of the nitrogen cycle, which we talked about in the last unit. They're important for food digestion in humans, so synthesis of vitamins, and that happens in our intestines. They're a source of antibiotics. They're used to treat chemical toxic chemical spills. So the bacteria convert these toxins to non-toxic CO2 and H2O or chloride compounds. This is cost effective, and the bacteria only grow as long as a toxic chemical is present. Harmful bacteria. So the modes of attack. So they attack, for one, because of their sheer numbers. It puts a strain on the host cell and tissues. Number 2, they destroy the host cell and tissues. Number 3, produce toxins. Harmful bacteria includes causes of pneumonia, strep throat, skin infections, causes of food and gasoline spoilage, causes of water pollution and plant rot, as well as destroying silkworms. For more information on bacteria, please read pages 340 and 346 in your biology textbook.